Hi, this is Dr. Jabali, and in this video, we're going to work on past papers from IB, in which focus more on the written answer questions such as describe, explain. So this is short answer questions as well as long answer questions, but they're all going to be focused just on topic 3.3, which is all about meiosis. So let's go with the first question here. It says, explain how error in meiosis can lead to Down syndrome. Here it's very important that you know what's the main error here. And the main error that we start with this is known as non-disjunction. This word, you will definitely get points for it. Not only do you get points for it, but you have to mention what non-disjunction is. And it means just to fail to separate chromosomes. In this case, if it is meiosis 1, you will actually say homologous chromosome. And if it's in meiosis 2, you will say that it's the sister chromatid. So it's very important that you mention the difference between the two. So, and usually where do these two happen? This one happens in anaphase 1, and this one happens in anaphase 2. So make sure that you link homologous chromosomes to anaphase 1 and sister chromatids to anaphase 2. They're both happening in meiosis. Now, when this fails to separate, what does this cause? This causes the sperms or the eggs that are forming to have, less or more chromosomes than the usual. And this is where you have to put like an example of a situation like this, and you can say about Down syndrome. But remember, just putting the word Down syndrome is not enough. You have to explain what Down syndrome is. And you can say that this is a trisomy, which means it's basically 3 chromosomes. And you gotta have to say where's location. 3 chromosomes found in the chromosome 21. Now, you can also mention about this, how did they fail to separate during anaphase? In this case, you can always mention about the spindle, how the spindle wasn't working, or how the centromeres did not split. So this is basically the general ideas that you have to write about Down syndrome when it comes to meiosis. So let's see what the mark scheme is working. So the first thing it says here is non-disjunction. Just mention that name, you get the first point. Second, you can, before I even continue, it says appropriate annotated diagrams. So that means, what I mean by annotated diagrams, you can put here anaphase 1, and you can have here anaphase 2, and you can draw it. But when you draw, you got to be very careful, because anaphase 1 is a separation of 2 of basically of the homologous. So that means it's 2 on this side, and then you can say that here you have this, and then eventually you can have that these 4 will go this way, and there's no separation going here. Anaphase 2, this will mean the sister chromatids. So this is going this way, this is going that way, right? And suddenly you can have 2 going this way and none this way. And then you have 1 going here, 1 going there. So you can see that the difference between anaphase 1 and anaphase 2. So you can always say that they are not going to be separated. The chromatids or the chromosomes, they don't go to the same, they basically go to the same pole. As you can see here, they're all going to the same pole simply because they were not separated. So it's non-separation of homologous chromosome during anaphase 1, as I have mentioned. You can say it's because the spindle did not work on attaching. And you can say non-separation of the chromatids in anaphase 2. So you can see homologous on anaphase 1 here. And you can see because of the centromeres, it did not divide. It can be that the gametes, the sperms formation, this is usually happening during meiosis, right? When you try to form these guys. Now, it's more common to see this happening with the egg instead of doing with the sperm. So most likely the woman will have malformation in the eggs and not the sperm necessarily. Then you can give an example, which is the Down syndrome, which is the extra chromosome in 21. And you can say, how does this happen? And you explain the gametes, how they receive an extra of the same type. So you can always explain how about the sperm having 23, while the egg is coming with 24. And you can show here that this becomes 47, which means it's 46 plus 1 extra chromosome. And they can say that this is extra of one of those 46. You can talk about the zygotes having 3 chromosomes, and that means they're trisomy. So that means in total 47. And that's pretty much it for the Down syndrome. So very important that when you're doing this essay, when it comes to Down syndrome and meiosis, the main words that they will look for is non-disjunction. It is homologous in anaphase 1, the chromatids in anaphase 2. It is giving Down syndrome as an extra chromosomal 21, which is trisomy. These are the main things that must fall in the mark schemes, and they show up in every mark scheme that talks about Down syndrome. So make sure to put these, plus the other ones, but these ones are the most common ones. So make sure to memorize these. Now the next question says, outline what occurs in a cell in the first division of meiosis. So basically they're asking you to describe meiosis 1. So when you talk about meiosis 1, you can also draw and then give annotations. You can get points for that. So the meiosis 1, we have prophase 1. We have metaphase 1. We have anaphase 1. Remember, you have to write the entire name. And then there is the telophase 1. You cannot abbreviate them by saying P1, M1. You got to get the full names written. Now, in the prophase, you can mention here that the nuclear envelope, if it disappears, you can say here that the most important thing is that the homologous chromosomes pair up. And this is where the crossover is actually happens. So these are the main things about prophase 1. And metaphase, you can say that the homologous, and I'm going to put CR here just as chromosomes, line up in the middle of the cell. And you can say that it was a spindle that line up them. And where does the spindle come from? The centriole. Then in anaphase, you can say that the spindle split the homologous chromosomes by separating them and drag them to each pole. And you can say separate them equally. And in telophase, you can say that the nuclear envelope appears and the chromosomes uncondense, and this you should get all the full points. You also can do the drawings. So when it comes to the drawings in this case, the prophase in this case, you can see like a dotted line, that will be the nuclear envelope broken down. And then very important that you show 4, like these line up together, and you can see the spindles starting to form from each side. That's prophase. But the most important thing is to definitely show basically a homologous right here. There's 3 of them here. The metaphase on this case, you have to show 4 in the middle because we're talking about homologous. And you have the spindle connecting them, and spindle connecting them, and you got the centrioles. When it comes to anaphase, you have to show 2 going, that means the homologous have been separated and each 2 is going to the opposite pole. And then the telophase, do not make 2 cells, still make a cell connected in the middle, and you have the nuclear envelopes with the chromosomes not visible anymore. That's how would you do the drawings for meiosis 1. So let's look at the mark scheme. Halves of the chromosome number produces haploid cells. So this was just explaining what meiosis is about, because meiosis again is all about going from a 46 to eventually going to a 23. So you're going from a diploid to become a haploid cell. Next, at the start of meiosis, each chromosome consists of 2 sister chromatids attached by a centromere. So this is the homologous chromosome that I was saying. You have here a sister, and here you have another sister. And these sisters are together. So basically you have 2 sisters, 1, 2, and they're connected, each one by their centromere right here in the middle. Now in prophase 1, you can say the pairing of the homologous chromosomes, as I mentioned. You can talk about the crossover that occurs. The crossover condensed by supercoiling, that means the chromosomes become visible. So every time we say the word condense, they become visible. Then in metaphase, the pair of chromosomes, homologous chromosomes, they move to the equator, that means in the middle. So it's the same thing. Then we can talk about anaphase, which the centromere does not divide. What really divides here is that the spindles push the chromosomes, in this case the homologous chromosomes, to each side. So that means the sisters are the ones that are going the opposite side of the poles. And then the telophase, you have the arrival of the centromere with the sisters at the opposite poles. And you can definitely mention here that the nuclear envelope doesn't condense. Now here's the crazy thing, is that here, even though the Mark scheme does not say telophase, the chromosomes uncondense and nuclear envelope doesn't get back up, that's the pain about IB. Sometimes they will not have there, even though if you write it, it is right. So in this Mark scheme, it will not give you the points. But they will give you points for annotated diagrams. But you gotta be careful. Do not mention meiosis 2 here because you're not gonna get any points to it because it says only the first division. So moving to the next question, it says here, explain the need for halving the chromosome number during sexual life cycle and how it is done. So when it says how it is done, that means we have to explain what meiosis looks like. And then when it says why do we halve this, that means why do we go from 46 to make 23? Why do the sperms and the eggs have to be 23, right? And the reason why is because when they come together, they turn into 46 and you're going back to being a diploid, which you go back to being human. But imagine if the sperm and the eggs, both of them were 46, then this would have been an issue because it would have come on 92, and that means this diploid here would have been completely a new species, and that is definitely not what the human is. Now, when it comes to how the meiosis is done, it's basically what I just explained in the previous slide about meiosis. You talk about the steps of it. Now, just to generalize what this meiosis looks like, you can say that meiosis has meiosis 1, in which you started with a diploid right here, and the diploid is usually 46, and then you go through prophase, metaphase, anaphase, telophase, all of them are the 1. And then you have a 46 and 46, and usually prophase here is where you have the crossover. And through all this process, you are using the word homologous chromosomes. So homologous chromosomes line up in the middle, homologous chromosomes separates, and so forth. Then you go through meiosis 2. In this process, again, you have the 4 cells coming out, and these cells can be known as gametes. Now the gametes, they can be sperm or they can be egg, and all of them should be half. They should be 23, so all of them should be a haploid of what was previous. To get there in meiosis 2, you have prophase 2, metaphase 2, anaphase 2, and telophase 2. Here you do not have any more crossovers. And here we usually say sister chromatids are lined up in the middle, sister chromatids are separating, and that's forth. So this is a very generalized, but of course you can go more in details about each part of the meiosis. So let's look at the Mark scheme. The first one says chromosome number is halved. As I mentioned, it did halve here. I said that it goes from 46 to 23. So the zygote can same number as the parents. So that's why you don't want the chromosome to double. Remember, it's when I said that you don't want to go to 92 because that's a new species. The process is meiosis. See, because it said the word how is it done. So here it says how it's done. It did not give you the name meiosis. That's the moment you can get a point by saying, oh, it's done by meiosis to get that point. Here we can say DNA replicates. Why? Because before you can start meiosis, if you remember the cell cycle, you have G1, S, G2, and then you hit meiosis. So for that, you have to do the DNA replication right here, in which you're making the copies. So you have to create the copies before you can even get to meiosis. The homologous pairs in chromosome in prophase 1, we can say that and they separate during anaphase 1 into the 2 cells. And after meiosis cells are haploid, that means after meiosis 2. But this one right here, you got to be careful because it's not after meiosis 1, it's after meiosis 2 that the cells are haploid. So this is a part of the Mark scheme. There might be a mistake here, but you can say that the final result of meiosis is a haploid. Here it says in meiosis 2, chromatids are separated. As I said, we use homologous chromosome for meiosis 1, chromatids for meiosis 2. As a result, we have 4 haploid cells coming out, which is true. There's 4 sperms or 4 eggs coming out, and each gamete is genetically unique. Now, why is this genetically unique? Is because of a crossover that happens in prophase 1. And then the nucleus crossover, as I mentioned, it also has an independent assortment, which is how the chromosomes in which direction they're going this way or that way. Fertilization results in the formation of a diploid, as I mentioned. That means if you have a sperm that is 23 going with an egg that's 23, when they come together, becomes 46, and that is known as a zygote. And that is when we start as a diploid and we start to divide and become us. So here it has some of these guys. Now, if you write the entire meiosis step by step, all the way from prophase 1 all the way to telophase 1, and you say what meiosis is about and how you're going from a diploid to a haploid, and talk about the crossover and mention about the homologous chromosomes before and then the sister chromatids on the second part, mention how there's a final 4 haploids at the end, then there's no chances of you missing out all this information. And of course, when there's a fertilization, there's a variation population, right? Because imagine if you're producing 4 sperms and you got those 4 eggs, there's so many portions. This sperm can go with this one or that one or this one. This sperm can go with this egg or this egg or this egg. You can see so many options. And this is just between, one production of meiosis from mom and dad. But imagine they do meiosis all the time, right? Especially the men. And we have billions of people doing meiosis. So you can see how much variety of sperms that we're producing every day, as well as the eggs that's coming every day from the woman. Now, moving to the next question says, describe the process of crossover. Now, crossover, the first thing you have to mention that it happens with the homologous pair. And you can say that they line up next to each other. And this is happening during what? Prophase 1. Remember, it does not happen in prophase 2, just prophase 1 of what? Of meiosis. So here you can also draw that they will allow you to do it. So I will draw here a sister chromatid with A, B, C, and then a sister here with A, B, C, because these sisters have to be exactly. And then I would put the homologous chromosome, which is another sister that looks the same, but it's now I changed the alleles to little alleles here. So these are 2 sisters. You can see this is the first, the second sister. But as together, the 4 of them, they are known as homologous pair. Or we can say that they are bivalent or tetrad. These are all names they can use them. Then if I want, I can show a crossover here where a crossover happens in between the 2 sisters, right? In between these 2. And how this will lead to 4 different types of variations of chromosomes. So this will be like A, B, C. This will be little a, big B, big C. And this will be big A, little b, little c, and then little a, b, and c. So you can see now I have 4 options. This is how I would draw, and then I would cross here and say the word crossover. So this is showing you that things can exchange, and you probably get points for it. So let's look at the mark scheme. First occurs during prophase 1. That's very important, during meiosis. Second, homologous chromosomes form bivalent, as I mentioned up. Breakage and rejoining of the chromatids. And that breakage and joining of the chromatids is basically here. There's a break that is happening where the A's are flipped. And then exchange of DNA between non-sisters chromatids. Very important that you say this. Now it's written in words the non-sisters, but if I show my drawing here, you can see that, right, this is going with the sister of the other one kind, so they're non-sisters. So the crossover is happening between these 2. In the drawing is showing you guys, if you want to write in words, then you must mention that the crossover is happening between non-sister chromatids. And that's pretty much for that. So now let's talk about compare. Now compare, in the old curriculum, it would have been give me the differences. But in the new curriculum, it means similar. But in this case, I'm going to take compare from the old system, and I'm going to look at the differences between them. So if this is the case, I would use the word contrast. So we're going to contrast mitosis and meiosis. So I'm going to make a very quick chart here that will help you out. And then you can use how many points it will come out of this. Now, there's so many differences between them, but I think that usually these questions can be 5 points and maximum 7 points out of this. So I'm going to give you around 5 to 6 of these, and if you write them, they're definitely going to hit one of the mark schemes. The first thing is that this one's going to be 2 identical daughter cells, while here they will be 4 different cells. Here we're going to say that we're going to come out with a diploid cell as a result, and here they will be haploid cells. We can say mitosis has just one process, that means one prophase, metaphase, anaphase, and telophase, while here you go to the process twice, right? 2 times. So that means you have prophase 1, metaphase 1, anaphase 1, telophase 1, and then you got prophase 2, metaphase 2, anaphase 2, and telophase 2. You can say mitosis is all about repair and growth. That's the reason why you do mitosis. And here is to produce gametes. In this case, we can talk about the sperms and the eggs. Now, here in prophase 1, there is crossover. And this causes variety in in the genes, right? In genetics. While this one, they are identical. So these are just examples of difference between mitosis and meiosis. Now, meiosis also has homologous pairs, in which in mitosis it doesn't have it. It only has the sister chromatids. You also can put the numbers here. Here starts with 46 and you end up with 46. Here you start with 46 and you end up with 23 chromosomes. So these are just some examples of mitosis and meiosis. So let's look now what are the mark schemes we have for this. The first one is mitosis has 1 cell division, meiosis has 2. So that's one of the things we said. My chromosomes number does not change, while this one you're going from diploid to haploid. Produced genetically identical, and these ones they're diverse, they're different. Separation of sister chromatids anaphase, while meiosis you have homologous during anaphase 1 and sister chromatids anaphase 2. Chromosomes has no crossover, meiosis has crossover in prophase 1. There is no formation of tetrads in mitosis, and there is a formation of tetrad synapses in meiosis. Mitosis produces cells to growth and repair, as I mentioned, while meiosis, it's all about the sexual cells. That means you're producing the gametes. Mitosis has 2 cell producers. Meiosis has 4 cell producers. Where I'm going here, mitosis, the daughter cells with both copies of chromosomes, does not occur. Here, meiosis, there's a random assert maternal, paternal. So that means, yes, it basically is switching around. So that means there's more chances of having different types of chromosomes coming up. Mitosis replication interface, meiosis replication interface 1. These are when they're just getting more detailed stuff. Here, mitosis has 4 phases, as you mentioned, and here has the same 4 phases, but it's actually twice. So by now, it's a lot more points. So just mention these 5 here or 6 here. You're already getting the full points because it's not going to go more beyond than that. So now we're moving to the next question. You describe the behavior of chromosomes in the phases of meiosis. So here is just, I'm going to do a drawing so you can see what I'm talking about. Meiosis here compared to meiosis 2. Now, the most important thing is what we're using. Here, we're always going to use the word homologous. Homologous chromosome. Always, to everything that you mentioned. Here, you're always going to use the sister chromatids. Now, of course, we have prophase 1, metaphase, anaphase, and telophase. So as well as here. But of course, 1 is in 2. Here, we got to mention the crossover that is happening, while here there's no crossover. Here, we're talking about the homologous being in the middle, right? While here, we're talking about the sisters being in the middle. Here, we're talking about the homologous separating. And here, we're talking about the sisters separating. And then here, we're forming 2 cells with 46. And here, we're forming 4 cells with 23 chromosomes instead of 46. So that's the separation between meiosis 1 and meiosis 2. So it's very important that you always remember the homologous chromosomes is from meiosis 1 and sister chromatids from meiosis 2. So let's look at the mark scheme. So here you can start by saying the interphase, the cell growth, the replication, right? The G1, S, G2, all this happen here. In meiosis 1, and what happens in prophase 1, DNA supercoils, it becomes condensed, nuclear membrane dissolves, the homologous pairs becoming form bivalent, and crossover occurs. Here, in metaphase 1, spindles from the centrioles attach to centromeres, and they line up in the middle. Anaphase, the spindles contract and split the homologous chromosomes, so they move to the opposite side of each other. Telophase, chromosomes decondense, nuclear envelope comes back, and the cell divides through cytokinesis to form 2 haploid daughter cells. In this case, it's still diploid 46 because haploid would have been 23. Interkinesis, you do not need to know, but this is just basically saying that it's a period between meiosis and meiosis 2, and there's no replication whatsoever. Then in meiosis 2, you say now chromosomes condense, nuclear envelope membrane dissolves, the centrioles move to the opposite poles. And see, there's no crossover here. Here you can say the spindles put who? Now the chromosomes, you see it's not homologous. It line up in the equator. And in phase 2, you can say the spindles contract and split who? The sisters. And they move the opposite poles. And telophase, the chromosomes decondense. That means they don't become visible. Even nuclear envelopes come back, and then the cells divide, and it makes 4 haploid daughter cells. So in the end, it's all about forming 4 haploid gametes, and all of them are genetically different from each other because of prophase 1 when crossover happens. So this is a very detailed one. Now, they're never going to ask this because if you look at this, this is a very large mark scheme, but this is a question that I pick up that you must know, and I usually put all of this together so you can have a sense of what meiosis 1 is about and what meiosis is about. And if you notice here, when it comes to meiosis 1, you can see that it's all about the homologous pairs. It's homologous all the way. While on the meiosis 2, it's all about the sister chromatids going on here. It's dividing, splitting, middle, everything going on. So make sure to make that distinction between the two. Moving to the next question, it says, explain how meiosis results in effectively infinite genetic variety. So we're talking about why is that we are so different? How come our genetics are so different? And in the gametes through the crossing over. So basically, I'm going to have to explain how crossover happens in prophase 1 and the random orientation that happens in metaphase 1. Now, crossing overs that we told you about it's what I just draw in a few slides before, where you can have this being A, B, and C. This is little a, little b, little c. And then from the crossover that usually happens between the non-sisters, what happens is you get A, B, C on the first one, little a, big B, C, and then big A, little b, C, and then little a, little b, C. And now you can see that each one of these is a gamete or a sperm or an egg. So this is one sperm, this is one sperm, and this is one sperm, and this is one sperm. So these are 4 different types of gametes, and they all have a different type of genetic material. So that's what makes the genetic variety already happen. The second part is that when they are lined up in the middle here, like this, they line up in the middle by chance. It's not like they have, oh, this number 1 has number 2, number 3, number 4. They don't have this order. And the way they separate from each other, it means this might come this way, and this comes this way, and then on another cell, it might be that this one goes here, or this goes here, or this one decides to go here. This might want to go with this one, and these two might go that way. It's random. It's all the random orientation. They don't have a very organized order sense in the sense of saying that always the left goes to the left and always the right goes to the right. It's all by random. So by having these random, what does it mean? That means in the end, you can have those 4 sperms or eggs. By random chance, they don't know, maybe this guy will come here, or this will come there, or this will come there. We don't know. It's all by chance, depending on where these chromosomes were located during my metaphase and how they were separated in anaphase. So look at the mark scheme. Says during anaphase 1, homologous chromosomes separate such that they each result in a daughter cell and contains a chromosome of either maternal or paternal. So this is just saying that you're separating them during anaphase. The orientation of these homologs in metaphase 1 is random, so we don't know how they line up in the middle. They just do line up. And there's an equal probability that the daughter cell having either a maternal or paternal, so we don't know which one she's going to pick from. As a human have a haploid number, this means that there is 2 to the power 23 potential gamete combinations. So that means we have about over 8 million combinations to get different combinations of gametes together. Now, the crossing over that happens in prophase, that has to do with coming with new chromosome combinations, which I just did here on the back on the drawing. As you see, these were 4 different new ones combinations that we had here. So that way, it's all exchanged during crossover. So it's an infinite genetic variability in this case here. And other sources of genetic variation include random fertilization. That means you don't know which sperm is going to go with which egg. There can be DNA mutations, chromosomes mutations, or even non-disjunction. So from here, it's a full sentence writing, but if I had to put in words that you must focus, the first thing you will talk about is during anaphase, how the homolog separates. This is all by chance. The second thing is the orientation homologs in metaphase is always random. So that means there's equal chances that each parent will go in one way. Also, we talk about the combinations of the gametes, which is about 2 to the power 23. We also talk about the crossover that happens in prophase 1, how it gives you different types of variabilities of chromosomes. And then other variations such as random fertilization, DNA mutation, chromosome mutation, and non-disjunction. So these are the main things that you might have mentioned if you want to explain how meiosis gives effective infinite genetic variety. And that's pretty much it for 3.3. Here are some extra questions that might virtually comes up on IB exams. This can be found in the lecture of 3.3, so you'll be able to answer these questions too. And this is pretty